in DNA replication is the localized denaturation of that DNA molecule, meaning that at the origin of replication, that double-stranded DNA molecule will have to untwist. So it will have to become single-stranded. And so what helps in the localized denaturation of that DNA molecule is that enzyme called DNA helicase. So DNA helicase help to break those hydrogen bonds and cause the DNA to be denatured right at the origin of replication. Okay? So we said that untwisting of that DNA molecule uses energy, and that energy comes from ATP. Right? So the hydrolysis of ATP releases energy which is used by the DNA helicase in untwisting the DNA molecule. Okay? So after denaturation, what takes place? All right? We said that enzyme, which is called DNA primase, is responsible to make a short oligonucleotide, which is called a primer, that will anneal to both strands. So it is from that primer where that primer is attached. All right. So if you look at it this way, so you have the denatured DNA molecule here. All right. The primer is a short piece of oligonucleotide that anneals right there and right here. And so thereafter, the DNA polymerase will recognize where that primer is and start to build a complementary strand in this direction and in that direction until it is all complete. Okay, so that is the role of the DNA primase that actually binds to the helicase and the denatured DNA, and its role is to make that RNA primer that will anneal to both strands, and it is from there where polymerization will take place. All right. This diagram here outlines the different steps. All right. So it says right here at number 1, replication origin. So this is the origin of replication. So right here is the origin of replication where denaturation will take place. All right. So at the first step, the initiator protein binds to the replication origin. So what you see here in the blue is the initiator protein. All right. So that helps to recognize the origin of replication, and it binds to that DNA molecule right there at the origin of replication. Okay. The next step is you have DNA helicase binds to the initiator protein. All right, right there. So the DNA helicase binds to the initiator protein. So remember that the initiator protein helps to recognize the origin of replication, and it attaches itself right there to the origin of replication. The DNA helicase, remember, will be the one to untwist that DNA molecule, as observed here in step number 3. It says helicase unload onto DNA. And what is it doing? It is denaturing that DNA molecule right there, as seen in this diagram. So the DNA helicase is the one that is breaking those hydrogen bonds and denaturing that DNA molecule right there at the origin of replication, creating what is called a replication bubble. In step number 4, the helicase denatures the helix and binds with the DNA primase to form the primosome. All right. So here you have the DNA primase. And remember, what's the role of the DNA primase? It's to make that RNA primer. which will anneal to both strands, and from there polymerization will proceed. So the primase synthesizes the RNA primer, which is extended as DNA chain by DNA polymerase. So here you have the RNA primer. So this right here is the RNA primer. And it is from this point whereby polymerization will proceed in this particular direction. And of course, that polymerization is catalyzed by DNA polymerase. All right, so this is a process that takes place in both directions, all right, until the entire DNA molecule is replicated, okay? The initiation of DNA synthesis involves the synthesis of a short RNA primer catalyzed by primase. The complex of the primase, helicase, and perhaps other proteins with the DNA is called the primosome. Right? So the primosome is comprised of all those, the helicase, the primase, and some other proteins along with the DNA molecule. The primer nucleotide serves as a substrate for the action of DNA polymerase. It simply means that the DNA polymerase will recognize where that primer is annealed and knows exactly where to start polymerization. All right, so the primer is extremely important, and only after it anneals to both strands, then DNA polymerase can come and extend that complementary strand. So short DNA fragments called Okazaki fragments remove the RNA primers thereafter. The new DNA is made in the 5' to 3' direction. Take note of that and always remember that. So that new DNA molecule, all right, that complementary strand is built in the 5' to 3' direction. So if we look at this molecule here, let's focus on this bottom strand. The template strand says 3', all right? And so the complementary strand will be the 5'. This is what we see right here. So it will be built in the 5' to 3' direction, this way. My question to you is, what is this red portion here? What is this red portion? What is that? What do you think that short line in red represents?
Sir, isn't that the RNA primer?
Exactly. That is the RNA primer, right? So it is that RNA primer that will anneal first to this template DNA strand at the bottom, and from that RNA primer, then the complementary strand is built in the 5' to 3' direction. Now, take note of the strand at the top. Here you have the 3' end, all right, and this is the 5' end of that template strand. So, we said that DNA is built in the 5' to 3' direction, so this one will be built in this opposite direction. All right, I hope you guys are seeing it. And what you see here is the primer. This is the primer. And so this will be built in this direction, opposite to the other strand. What do you think this portion is, this circular right here? All right, this is what you see right here, shaded. All right, that would be the helicase that is helping to untwist that DNA molecule. All right, so it aids in untwisting the molecule and allowing DNA replication to take place. Now, I want you to visualize this, You have a molecule that is untwisting as it moves along. This bottom strand, the the complementary strand is being built towards the replication fork. Remember, this is called a replication fork. So that will have absolutely no issues moving along, because as the molecule is denaturing, that complementary strand will be building. However, that is not the case in the other strand at the top. In the other strand at the top, there has to be a waiting period. So you have to wait for that DNA molecule to denature before that RNA primer anneals and then builds in the opposite direction from that replication fork. This is very important. This, what I'm explaining, is very important because we'll come to what is called a leading strand and a lagging strand. So in this particular case of what you see here in this diagram, the leading strand would be the one at the bottom, because that would be leading the replication. As the molecule is being denatured, that complementary strand is building continuously. But in the strand at the top, it is called a lagging strand because it has a waiting period. It has to wait for the molecule to denature, then for the primer to anneal before it can build in the opposite direction. So that is called a lagging strand. All right? Questions? Lourdes, go ahead.
Good morning. I have a question.
Yeah, go ahead.
So DNA would always be the leading strand, and then the top would always be the lagging strand.
Sorry, Lourdes, I didn't get the first part.
I was asking about the DNA replication. Would it always be that whenever it's being replicated, the bottom portion would be the leading strand and the bottom top portion would be the lagging strand? Would that be the case every time?
You can't go by top and bottom, because, for example, if I do like this, I label it like that. It simply means that this complementary strand that is being built here this would be the 3'. All right? And remember, we said that DNA is built in the 5' to 3' direction. So here, the complementary strand here, this complementary strand here would be the 5', which means that this would be built in this direction. So in this particular case, the top would be the leading strand, while this bottom that is building in this direction would be the lagging strand. So it doesn't have to do with top or bottom, Lourdes, but rather where you have these label 3', 5'. That will indicate which one is the leading strand and which one is the lagging strand. Is that clear?
Yes, it's clear. Thank you.
Okay. Any other questions? So let's look at DNA replication in eukaryotes. Remember, we said that DNA replication in prokaryotes is relatively simple because we're talking about one circular DNA molecule, which proceeds in both directions until you reach the terminus and you end up with two molecules. DNA replication in eukaryotes. It's similar as in prokaryotes. However, the added complication is that DNA is distributed among many chromosomes in eukaryotes. In case of human beings, you have DNA distributed along all 46 chromosomes. In each cell division cycle, each of these chromosomes must be faithfully duplicated and a copy of each distributed to each of the two progeny cells. All right, so remember mitosis will end up with two daughter cells that are diploid and are identical to the parent cell. All right. So if you can recall the cell cycle, you have G1, S, G2, and M. All right. The DNA replicates during the S phase, and the progeny chromosomes segregate into daughter cells during the M phase. All right. So this right here is the entire cell cycle. This is a review of your cytology course from 2 semesters ago. So DNA replicates during that S phase. All right. And you have growth and preparation for cell division in the G2. And then you have mitosis taking place. And then it completes with cytokinesis. All right. Let's look at the molecular details of DNA synthesis in eukaryotes. Nucleosome. Anybody recall what's a nucleosome? A nucleosome would be there you have proteins, which are the histones. All right, so these are proteins, which are histones. And you have the DNA molecule that comes and wraps itself several times around these histones. Right? And that is how eukaryotic DNA is packaged within that nucleus with the aid of these histones. All right. So each one of these is a nucleosome. All right. So a nucleosome comprises of that histone protein along with DNA wrapped around it. All right. So replication of the eukaryotic chromosome must involve the replication of the DNA and of the histone core of the nucleosome, as well as doubling of the non-histones, right? So whenever you have DNA replication, it is not only the DNA molecule that must replicate, but rather you have to have those histone proteins that also need to replicate, because it will need, the DNA will need something to wrap around. So everything must replicate. When you have DNA replication, you have the DNA molecule that is replicating, you have the histone proteins as well as the non-histone proteins. All right? The sequential steps described for the DNA synthesis in prokaryotes also occur for DNA synthesis in eukaryotes. Namely, 1, denaturation of that DNA double helix, and then the semi-conservative, semi-discontinuous replication of the DNA. We spoke about semi-conservative, and we know what it means, right? Semi-conservative. Semi-discontinuous is what I explained a while ago when I spoke about leading strand and lagging strand. So the leading strand replicates continuously because it is moving towards the replication fork. However, the lagging strand replicates semi-discontinuously because it has to wait for denaturation to take place because it is moving in the opposite direction of that replication fork. All right? So these are two terms that you need to understand what they mean. Semi-conservative, if you remember this diagram here, this is what semi-conservative means, that it conserves one of the strand from the original DNA molecule, and then it uses that as a template to build a complementary strand. So you end up with, after one round of replication, you end up with 2 DNA molecules. Each of them have one strand from the original DNA molecule and one newly synthesized strand. Okay? So that is semi-conservative. And semi-discontinuous has to do with that leading strand and the lagging strand. So remember that the leading strand will be replicating continuously, while the lagging strand will be replicating semi-discontinuously. All right, so semi-discontinuous replication. This was what I explained earlier. So that first example that I used, we noticed that the strand at the bottom was the leading strand, but take note of this diagram that I have here. It's opposite, and it all has to do with the labeling of these strands used as template to build a complementary strand. What you need to remember at all times is that DNA, and when I say DNA, I'm referring to that complementary strand that is being built, is always built in the 5' to 3' direction. So take note of what is happening right here. This complementary strand at the top. See, 5' to 3' direction. And it is moving towards the replication fork. So as helicase is untwisting this DNA molecule, that complementary strand is being built in that 5' to 3' direction. So that strand is being built continuously. Now the strand at the bottom, because it has to be built in the 5' to 3' direction, has to wait. All right? And so it is built in the opposite direction of that replication fork. And it is being built by what is called Okazaki fragments. So each one of these fragments here are the Okazaki fragments. All right? And these fragments will eventually replace the RNA primers that were annealed to that DNA molecule. All right? So initiation of DNA replication, if there was only one origin of replication per chromosome, the replication of each chromosome would take many hours, and gestation would be many years instead of just 9 months. So in the case of eukaryotes, all right, in the case of eukaryotes, there are many origins of replication. In the case of prokaryotes, all right, we're talking bacteria, there is one origin of replication. But in the case of eukaryotes, there are many origins of replication. DNA replication is initiated at many origins of replication throughout the genome. At each origin of replication, the DNA denatures and replication proceeds bidirectionally in both directions. And the DNA double helix opens to expose single strands that act as templates for new DNA synthesis. Eventually, each replication fork will run into an adjacent replication fork. And that is depicted right here in this diagram. All right. So in that diagram at the top, you notice that it is depicting 3 different origins of replication. And so DNA replication will be occurring at all points. And eventually, each replication bubble or each replication fork will run into another replication fork until you end up with what we see here at the bottom, with 2 daughter DNA molecules. All right. Take note that these 2 daughter molecules have 1 strand from the original DNA molecule, which is the one in dark blue, and 2 strands, newly synthesized strands, which is the one in light blue. All right. So this is how it would operate in the case of eukaryotes, where you have several origins of replication. So it proceeds bidirectionally until one replication fork runs into the other replication fork, and until the entire DNA molecule is duplicated. Okay. So because A and T base pairs have 2 hydrogen bonds, it's easier to disrupt than GC base pairs with 3 hydrogen bonds. You learn that from chemistry, right? So 3 bonds are more difficult to break than 2, and 2 are more difficult to break than a single bond. Single strand binding proteins, which are abbreviated SSB. All right, so SSB stands for single strand binding proteins, not Social Security Board. So these proteins prevents the DNA from renaturing. And these SSB proteins stimulate polymerase activity. So basically what happens? You have the DNA helicase that denatures that DNA molecule. While during that period of time, when it is being denatured, the SSB proteins are the ones that help keep it that way until that complementary strand has been built. If it weren't for the SSB proteins, as soon as helicase denatures that molecule, before the primers could anneal, it would renature. All right, so the role of the SSB protein is to prevent the DNA from renaturing before the complementary strand has been built. So this diagram here outlines basically everything and all the key players in this replication process. So you have here the helicase unwinds this DNA double helix right there. These what you see here in black are the SSB proteins that prevents renaturation from occurring even before complementary strand has been built. Here you have this is the primase that makes the primer. What you see here in green is the RNA primer. All right. In the case of the leading strand, only one primer is required. All right, because that is moving in the direction of the replication fork. However, in the case of the lagging strand, it will have to make several primers. As that DNA molecule is untwisting, it makes a primer that anneals there, and then it builds in the opposite direction of the replication fork. All right. So from the primer, this has been built in this opposite direction, and of course, these segments are called Okazaki fragments. All right. So in this particular case, the bottom strand is the lagging strand, while the top strand is the leading strand. All right. Okay. So this outlines what takes place during DNA replication. What I'll do now is show you guys a video that talks about DNA replication.
DNA replication begins at a specific point in the DNA molecule called the origin of replication site. Initially, the enzyme helicase unwinds and separates a portion of the DNA molecule, after which single-strand binding proteins react with and stabilize the separated single-stranded sections of the DNA molecule. The enzyme complex DNA polymerase engages the separated portion of the molecule and initiates the process of replication. DNA polymerase can only add new DNA nucleotides to a pre-existing chain of nucleotides. Therefore, replication begins as an enzyme called primase assembles an RNA primer at the origin of replication site. The RNA primer consists of a short sequence of RNA nucleotides, complementary to a small initial section of the DNA strand being prepared for replication. DNA polymerase is then able to add DNA nucleotides to the RNA primer and thus begin the process of constructing a new complementary strand of DNA. Later, the RNA primer is enzymatically removed and replaced with an appropriate sequence of DNA nucleotides. Because the two complementary strands of the DNA molecule are oriented in opposite directions and the DNA polymerase can only accommodate replication in one direction, two different mechanisms for copying the strands of DNA are employed. One strand is replicated continuously toward the unwinding, separating portion of the original DNA molecule, while the other strand is replicated discontinuously in the opposite direction with the formation of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer. As the Okazaki fragments are synthesized, The RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
Okay, I have to leave now. So on Thursday, we will continue our discussion on transcription and translation, and that will complete the entire process of the central dogma of molecular biology. All right, any questions before we leave? All right, none? Okay, guys, take care and enjoy the rest of your day.